CICU devices.
Rhinochill device. Ultra-early brain cooling in cardiopulmonary resuscitation. Therapeutic hypothermia, currently referred to as targeted temperature management, has long been investigated as a neuroprotective strategy in patients resuscitated from cardiac arrest. This is based on the principle that reducing brain and systemic temperature can attenuate ischemia-reperfusion injury, decrease cerebral metabolic demand, limit cytotoxicity, and mitigate secondary brain injury. Traditionally, hypothermia is induced after return of spontaneous circulation using surface or intravascular cooling methods, but these techniques are often slow to achieve target temperatures and rarely begin until hospital arrival. Recent evidence has shifted clinical practice toward strict fever prevention rather than routine deep hypothermia. Nevertheless, the concept of ultra-early and selective brain cooling, particularly when initiated during or immediately after resuscitation, remains an active field of investigation due to its potential to improve neurological outcomes. The Rhino-chill intranasal cooling system was specifically designed to address this unmet need. It is a portable, non-invasive transnasal evaporative cooling device intended to induce rapid and preferential brain hypothermia. The system delivers perfluorohexane as coolant, together with oxygen or air into the nasal cavities, where the liquid rapidly evaporates. Perfluorohexane is used in the Rhino-chill system because its physicochemical properties make it highly efficient for rapid heat transfer during evaporative intranasal cooling. It has a low boiling point and high vaporization enthalpy, so when aerosolized into the nasal cavities and allowed to evaporate, it absorbs a large amount of heat from the mucosa and adjacent structures, promoting fast brain cooling. This evaporation extracts heat from the highly vascularized nasopharyngeal mucosa and the skull base, promoting direct cooling of the brain with secondary and more gradual reduction of core body temperature. The anatomical proximity of the nasal cavities to the anterior cranial fossa and the cavernous and pharyngeal venous plexuses provides a highly efficient heat exchange interface. This mechanism allows Rhino-chill to combine three modes of heat transfer: evaporation, convection, and conduction, resulting in faster cerebral cooling than is typically achieved with surface-based techniques. Importantly, the system does not require vascular access, infusion of cold fluids, or placement of external cooling pads. From a practical perspective, Rhino-chill was engineered for use in both pre-hospital and in-hospital environments. The device consists of a portable control unit, a single-use cooling catheter with dual nasal cannulae, and a supply of inert perfluorocarbon coolant in disposable bottles. It can be operated on an internal battery or external power sources, and uses either medical oxygen or breathable air as the driving gas. These characteristics make it suitable for use by trained emergency medical personnel in ambulances, helicopters, emergency departments, and intensive care units. Clinically, the system is intended for initiation of cooling during ongoing cardiopulmonary resuscitation, or immediately after return of spontaneous circulation in patients with cardiac arrest. Use of the device requires a protected airway and assisted ventilation, and standard contraindications to hypothermia and intranasal instrumentation must be respected. The safety profile of Rhino-chill has been favorable across multiple clinical settings. Most reported adverse events are local and mild, including minor epistaxis, transient nasal mucosal irritation or discoloration, and rare cases of periorbital subcutaneous emphysema. Serious device-related complications, like pneumocephalus, have been very uncommon. A substantial body of experimental and clinical research has evaluated the feasibility and physiological performance of intranasal evaporative cooling. Early studies demonstrated that Rhino-chill could achieve meaningful reductions in estimated brain temperature, often exceeding the decrease observed in core temperature during the initial treatment phase. Subsequent studies in emergency department and intensive care unit settings confirmed that 1 hour of treatment typically produces a decrease of approximately 2 degrees Celsius in tympanic temperature and about 1 degree Celsius in core temperature. The PRINCE study evaluated the use of the device in the intra-arrest pre-hospital setting. It demonstrated that Rhino-chill can be initiated during active cardiopulmonary resuscitation without interfering with chest compressions, airway management, or advanced life support procedures. But no survival beneficial was observed. Finally, as of 2026, the PRINCESS-2 trial is ongoing, with inclusion criteria refined to defibrillable rhythms only, and will compare pre-hospital Rhino-chill cooling versus conventional treatment.
If you've enjoyed our video, please press the like button and subscribe to support us so we can continue creating quality content like this.